welcome to lecture number 5 in our nervous tissue unit. So in lecture number 4, we went over the neuron action potential. In lecture number 3, we went over the resting membrane potential. Today we're going to finish the action potential, and we're going to go over the synaptic transmission. So remember the synapse here, you barely see it right there. Neuron A is the presynaptic neuron, neuron B is the postsynaptic neuron. And in between those is a tiny little space between the axon terminals and synaptic end bulbs and the dendrites of the postsynaptic neurons. There's a little space there. That's called the synapse. So you can also see it right there. That's the synapse. So you have the presynaptic neurons. This is the synaptic end bulb right here. And this is this is the dendrites of the postsynaptic neuron. So we got to find a way. So the action potential travels down the axon into the synaptic end bulb, and then that neuron needs to pass the message or the impulse to the postsynaptic neuron, but it's got to cross that space. So this is where we're going to be using neurotransmitters. All right, so a little review. So remember, the resting membrane potential of negative 70 millivolts. It's maintained primarily through the sodium potassium pumps, which is continuously using ATP energy to pump out 3 sodium ions and pump in 2 potassium or K plus ions. That's what helps maintain that negative 70 millivolts. Also remember, there are things like proteins, amino acids, and organic phosphates inside the neuron, which help to keep it negative inside. Now, once that neuron starts to be stimulated, either by neurotransmitters with ligand-gated channels, and that would be these guys right here, or mechanically gated channels being stimulated by some sort of physical stimulus. And remember, that's a graded potential. So it allows usually sodium to start leaking into the neuron. And if enough sodium leaks into the neuron to reach the threshold of negative 55 millivolts, boom, that's when voltage-gated sodium channels are going to open, and positively charged sodium is going to rush into the neuron, causing it to go from negative 55 millivolts, the threshold, to positive 30. Remember, this is where voltage-gated sodium channels close shut, and voltage-gated potassium channels open up. And then that's going to cause potassium to rush out of the neuron, positively charged potassium to rush out of the neuron, and that's what's going to bring it back down. This is called repolarization. And for a brief little millisecond, it's going to be below the resting membrane potential of negative 70 millivolts, and that's called hyperpolarization. And then the sodium potassium pumps kick in and return it back to the resting membrane potential. So remember, when it's going up, that's depolarization, and when it's going down in charge, that's the repolarization phase. All right, so it's like the picture I had 2 slides ago, just there's 3 of them instead of just 1. All right, so remember the action potential, the nerve impulse is going to travel down the axon. Now, when it's in the axon, remember, it's due to the inflow of Na plus or sodium ions into the neuron. So remember, when it reaches negative 55 millivolts, those voltage-gated sodium channels are going to open and allow sodium to rush in. And that's what happens all the way down the axon. But now we've reached the synaptic end bulbs, and now things get a little different. It's kind of the same, but the only difference is now it's not voltage-gated sodium channels that open, but voltage-gated calcium channels that open, causing calcium to rush into the neuron from high concentration of calcium outside the neuron to low concentration of calcium inside the neuron. And remember, when the calcium ions come into the synaptic end bulb, that's going to cause these synaptic vesicles filled with neurotransmitters to release their neurotransmitters into the synaptic cleft. Those neurotransmitters are going to migrate, diffuse across the synapse, across that space, going to bind with those ligand-gated channels on the postsynaptic neuron. Remember, these are the dendrites right here. And when those neurotransmitters bind with the ligand-gated channels, it causes them to open up. And that's what allows sodium in the postsynaptic neuron to start diffusing into the neuron, into the dendrites of the next neuron. And that's depolarization. And remember, if it gets depolarized enough by excitatory stimuli, excitatory postsynaptic potential, and it reaches the threshold of negative 55 millivolts, the next neuron will fire an action potential. All right, before we dive into synaptic transmission, I wanted to do a little brief review on the action potential. All right, so remember, the action potential is going to travel down the axon, the nerve impulse, and it's going to reach the axon terminals and then the synaptic end bulbs. This is going to cause the release of neurotransmitters into the synaptic cleft, the space between the presynaptic neuron and the postsynaptic neuron, the dendrites and the synaptic end bulbs, a little space there. So neurotransmitters have to cross that space. So they cross the synaptic cleft, and they're going to bind with these Receptors on the dendrites of the postsynaptic neuron. Remember, these are called ligand-gated channels because they open and close due to specific neurotransmitters. All right, so since there's neurotransmitters binding with those ligand-gated channels, they're going to open up. And that's going to allow sodium Na plus ions to start creeping in to the postsynaptic neuron. It's going to start depolarizing it, making it less and less negative. It's starting to inch closer and closer to the threshold. Now, if enough sodium enters the postsynaptic neuron to take it to the threshold of negative 55 millivolts, boom, that's where you're going to have an action potential. So if it reaches negative 55 millivolts due to enough stimulation, excitatory postsynaptic potential, then it's gonna reach the threshold and it's gonna cause this voltage gated sodium channel to open up. When that happens, sodium ions are gonna rush into the neuron from high concentration to low concentration, these positively charged sodiums through the voltage gated sodium channel. And that's what's going to cause it to peak at positive 30 inside the neuron. Well, now that it's positive inside the neuron, it's a chain reaction. Now, it being positive is going to stimulate this voltage gated sodium channel to open up, and it's going to cause the next round of sodium coming in, diffusing from high concentration outside to low concentration inside. So now it's going to be positive in here, positive 30. And that's going to cause the next voltage gated channel to open up, allowing more of these sodium ions to rush into the neuron. And this happens all the way down the axon. One after another, these voltage gated sodium channels open and allow sodium to diffuse into the neuron. That's what an action potential is, the opening of voltage gated channels all the way down the axon. Now remember, it's this inflow of sodium ions that contributes to the depolarization of the neurons. Remember, you have the resting membrane potential of negative 70 millivolts. It starts to get stimulated. If it reaches negative 55 millivolts, the threshold, boom, you got the action potential. Remember, this going up, that's depolarization. But now we got to get that graph back down. We got to get it back down very quickly, back to that resting membrane potential so the neuron can fire another action potential. And this is how it happens. So, now that we have it, now that it's positively charged inside the neuron, positive 30, that's going to open up this other voltage gated channel, and that's the voltage gated potassium channel. So now it opens up, it's positive on the inside. It's going to allow potassium to rush out of the neuron, positively charged potassium, from high concentration of potassium inside to low concentration of potassium outside. And the movement of all those potassium ions is good. That's what makes it positive on the outside again. This is repolarization. So now, once again, it's positive on the outside, it's negative on the inside. All right, so now we've repolarized the neuron. We got it back to positive on the outside. We got it back to negative 70 millivolts on the inside, but we have not restored the ion distribution because look at all the sodiums on the inside. They're supposed to be on the outside. Look at all the potassiums on the outside. They're supposed to be on the inside. Well, how are we going to get those flip-flopped? Well, remember, it's going to be the work of this guy right here, the sodium potassium pump. He's going to continuously pump out 3 sodium ions from the inside to the outside. He's going to pump in 2 potassiums. So the continuous work of the sodium potassium pump, now we have both our electrical conditions, negative on the inside, negative 70, and positive on the outside. But we also have once again a high concentration of potassium on the inside, low on the outside, and a high concentration of sodium on the outside and low concentration of sodium on the inside. So that is how a neuron, the action potential, the depolarization, repolarization. Now remember, those potassium channels are slow to close. So it briefly goes even below negative 70 millivolts. Remember, that's called hyperpolarization. And then the sodium potassium pumps kick in and bring it back to that resting membrane potential of negative 70 millivolts. All right, so now a couple things to determine how fast the action potential can travel down the axon of the neuron. So it's the speed of conduction, how fast it can travel or conduct down the neuron. And one type of conduction down the axon is called continuous conduction. And these are axons that are unmyelinated. So remember, myelin is the fatty covering around the axon. Remember, it's produced by oligodendrocytes in the CNS and Schwann cells in the PNS. So in continuous conduction, when there's no myelin on the axon, it's a step-by-step depolarization. So they say it's kind of like a domino. So the first sodium channel opens, voltage gated sodium channel opens, it causes the next one to open, and the next one, the next one, the next one. But an unmyelinated axon, because it's continuous conduction, it's got to travel the entire length of the axon. So this is going to be much slower. So unmyelinated axons, continuous conduction is slower than this one right here, saltatory conduction. So these are axons that do have myelin on it. Now remember, Myelin, in between the myelin, there's these little spaces called the nodes of Ranvier. And in saltatory conduction, the nerve impulse, the action potential, does not have to travel the entire length of the axon. It's able to jump from node of Ranvier to node of Ranvier. And since it's jumping over large segments of the axon, it is much, much faster. All right, so saltatory conduction, it was myelinated axons, much faster. Continuous conduction is unmyelinated axons, and it's much slower because the entire length of the axon needs to be traveled. The whole thing needs to be traveled. Nothing can be skipped. Another thing, two more things that can affect the speed of conduction. Diameter. The larger or the wider the neuron, faster the conduction. And also temperature. Higher temperature, the faster the conduction. So, I mean, that's just because things are able to diffuse faster. Molecules move faster when there's increased temperature, and they slow down when temperature gets slower. So that's one reason why if something's sore, like maybe your arm is sore, your knee is sore or something, you'll put ice on it. And that serves two purposes. It reduces inflammation. The inflammation is swelling that's pushing on some sensory neurons and causing it to hurt. But also the cold temperatures cause your action potentials to slow down. So instead of having a real high frequency of action potentials telling your brain this really hurts, it'll slow it down a little bit and your brain will say, that doesn't hurt quite so bad. And here's some pictures that show the difference between continuous conduction and saltatory conduction. So over here on the left, we have continuous conduction. You can see this axon right here has no myelin. And over here, this axon does have myelin. So it's going to be saltatory conduction over here. All right, so you can see continuous conduction. Here's right here. This is where the actual local depolarization is occurring. So this is where sodium is rushing in right here. And it's going to cause the next segment for sodium to start rushing in, which is going to trigger the next sodium channels and the next one, the next one. So in continuous conduction, it's got to travel the entire length of the axon. One sodium channel after the other gets stimulated to open up, causing the action potential. So you can see after 1 millisecond, it traveled this far. After 5 milliseconds, it traveled this far. And after 10 milliseconds, it traveled this far. All right, so that's continuous conduction. So it's got to travel the entire length of the axon. Now here's saltatory conduction over here. So you have your myelin right here. The spaces in between the myelin are the nodes of Ranvier. So this is where it can jump from node of Ranvier to node of Ranvier, allowing it to skip large portions of the axon. So this can be much, much faster. So you can see after 1 millisecond, it traveled like the same as continuous conduction. But after 5 milliseconds, it's traveled way over here. After 10 milliseconds, it's way down here, where in continuous conduction, it was just right here. So saltatory conduction is much, much faster because the action potential, the nerve impulse, is able to skip large regions of the axon, jumping from node of Ranvier to node of Ranvier. All right, so here's some key terms for your notes. Now, people always ask, hey, do I have to take notes? No, you don't have to take notes. I mean, if you have a photographic memory and this is just instantly, boom, gets stored in your brain, I mean, taking notes would be a waste of time. But if you do take notes, when the questions pop up on your Edpuzzle, they're going to be a lot easier to answer. Now, some people do like the lazy way, the shortcut of taking a picture of it with their phone and then having that picture to answer the questions. Is it really good for answering the question? It is. Is it as good to remember it as taking pencil and paper and actually writing it? It is not. So whenever you take a shortcut, it usually turns into the long cut, because now before an exam, you're going to have to study much more. You're going to have to cram because you didn't learn this stuff, because you didn't take the notes, and interact with the material. But anyways, so those of you that are taking notes, write this down. The synapse is basically just the junction between the presynaptic neuron and the postsynaptic neuron. The synaptic end bulbs in the presynaptic and the dendrites of the postsynaptic. Synaptic end bulbs, this is the enlargement or the widening of the axon terminal. So the axon, first you have the axon, it'd be like my arm. The axon terminals would be like my fingers. And if my fingers, they are kind of a little bit fatter at the end, but if they got even fatter, that would be the synaptic end bulbs. These are the axon terminals. This is the axon. This would be the synaptic end bulbs. Synaptic vesicles, those are like little bubbles called vacuoles. And those are what's holding the neurotransmitters. Those are the chemical messengers that are going to cross the synaptic cleft, the space between the two neurons. And synaptic cleft, obviously, that is the space between the synaptic end bulbs of the presynaptic neuron and the dendrites of the postsynaptic neuron. Neurotransmitter receptors. These are the little gates, the little receptors on the dendrites of the postsynaptic neuron. So the neurotransmitters are going to diffuse or cross the synapse. They're going to strike the receptors on the postsynaptic neuron. Remember, those are called ligand gated channels. And the neurotransmitters are going to cause that ligand gated channel to open up and allow sodium to leak into the neuron. And that's what starts to stimulate it. And neurotransmitters, those are the chemicals that cross the synapse in order to stimulate the postsynaptic neuron. All right, so here's a picture of a synapse. So this is the far away view. So here's the presynaptic neuron right here. Here's its synaptic end bulb. Here are the dendrites of the postsynaptic neuron. And right here, there's a synapse. And here's a close-up view of it over here. All right, so this would be like the axon terminals of the presynaptic neuron. So remember, as the action potential is propagating or moving down the axon, that is caused by voltage-gated sodium Na plus channels opening. That causes sodium to rush into the neuron, which is going to depolarize it from the threshold of negative 55 millivolts, and it's going to shoot up to positive 30. That's what an action potential is. And that's what's happening the entire length of the axon. But now when we get to the synaptic end bulb, something different is going to happen. So as the action potential travels into the synaptic end bulb, now it's not sodium ions anymore. Now we're switching to calcium ions, Ca2 plus. So when the action potential reaches the synaptic end bulb, that's going to cause the voltage gated calcium channels to open up. And calcium is going to rush into the neuron from high concentration to low concentration. When this happens, the influx of calcium is going to cause these guys right here. These are the synaptic vesicles, little bubbles, vacuoles, filled with those little dots. Those are the neurotransmitters. So that when the calcium comes in, that tells the synaptic end bulb or the synaptic vesicles to fuse with the synaptic end bulb and release all the neurotransmitters through exocytosis, which you will not need to know, into the synapse or the synaptic cleft right here. And all these little neurotransmitters are going to move or diffuse across the synapse, the space, the synaptic cleft, and it's going to bind with these little receptors on the dendrite of the postsynaptic neuron. Remember, these are called ligand gated channels because they open or close due to a specific neurotransmitter. And when these neurotransmitters bind with these ligand gated channels, they open up. When they open up, they allow sodium to start creeping into the neuron, positively charged sodium, usually. Well, in this case, it's almost always positively charged sodium coming to the neuron. That's going to start to depolarize that neuron. It's going to start getting it off that resting membrane potential of negative 70. It's going to start making it less negative or more towards the positive. And remember, if that thing gets stimulated enough and it reaches that magic number of negative 55 millivolts, boom, you'll have an action potential. So this is what happens at the synapse. The action potential reaches, voltage gated calcium channels open, positively charged calcium enters the synaptic end bulb. That tells the synaptic vesicles, go ahead and release your neurotransmitters into the synaptic cleft. They travel across the synapse, bind with these receptors on the dendrites of the postsynaptic neuron, ligand gated channels. They open and allow sodium to start coming in, which is going to start depolarizing the postsynaptic neuron. All right, it's basically the same as the last slide, just in word form. Now remember, you do not have to copy these words down word for word. Feel free to put it in your own vernacular or your own language, kind of like text messages. You can shorten this, put it in language that you can understand. All right, so number one, so let's see if I forgot anything. So the action potential reaches the end bulb. Remember, that was voltage gated sodium channels opening and closing all the way down the axon. That's how the action potential traveled down the entire axon. And remember, to repolarize the neuron down the axon, it was the opening of voltage gated potassium channels that allowed potassium to exit. And that's what caused it to repolarize back down to negative 70. All right, but anyways, when the action potential reaches the synaptic end bulbs, that's going to cause voltage-gated calcium channels to open and calcium to flow into the synaptic end bulb. So far, I didn't forget anything. The calcium triggers the release of neurotransmitters. Oh, by exocytosis. I didn't even know that was going to be in here. I doubt you need to know that. That's a way that cells can rapidly get rid of or remove large amounts of substances like liquids or ions or anything. But I don't think you'll need to know that. The neurotransmitters cross through the synaptic cleft, the space between the two neurons, and it's going to bind to receptors on the postsynaptic membrane or the dendrites on the postsynaptic neuron. Remember, those are ligand gated channels because they open and close due to a specific neurotransmitter. Let's see. Ion channels open in the postsynaptic neuron or membrane or the dendrites, and that's going to allow the influx of Na plus sodium ions to come in. And that's going to start to depolarize the postsynaptic neuron. All right, so I don't know if you remember from the last lecture on the action potential, but remember, neurons can be excitatory or they can be inhibitory. So if they're excitatory postsynaptic potential, they depolarize the neuron and make it more likely to fire an action potential. But there's also inhibitory neurons, and their neurotransmitters, inhibitory postsynaptic potential, those actually repolarize the neuron and make it less likely to fire an action potential. So this is where it's talking about right here. If the neurotransmitter, remember it's usually GABA, GABA. If it's inhibitory, it's going to one way it inhibits the neuron is it causes chloride channels to open, and then chloride ions, which have a negative charge, will start diffusing into the neuron from high concentration of chloride to lower concentration of chloride, and that's going to, since they're negative, it's going to cause it to repolarize even more or even hyperpolarize, causing the neuron it'll be less likely to fire an action potential because it's getting further away from the negative 55, not closer to it. And if the neuron receives enough excitatory stimulation, excitatory postsynaptic potential, and it depolarizes that neuron enough to reach the threshold of negative 55 millivolts, boom, you're going to get a nerve impulse or action potential. And remember that process of adding up all the excitatory stimulation it receives and all the inhibitory stimulation it receives, it adds it all up. And if it reaches negative 55 millivolts, that's the threshold, you'll have an action potential. Remember that summation occurs at the axon hillock. So that's where the action potential will initiate or start.